Hello everyone. In this lecture, you will get to know about Aspergillosis. Aspergillosis is the fungal infection caused by the fungus Aspergillus. The Aspergillus fungus is also known as green fungus. The fungus Aspergillus is commonly found in soil. It shows saprophytic mode of nutrition. That means it obtains its nutrition from the dead and decaying matter. It is a spore-forming fungus, reproduces asexually by producing the spores, which are also called conidia. These spores are usually found in air and soil. The spores germinate in the presence of moisture, warmth and nutrients and produce the vegetative form hyphae. The Aspergillus fungus is classified in division Ascomycota. The important species are Aspergillus fumigatus, niger, flavus, nudulens and clavens. There are some good features about Aspergillus. It has been used for the commercial production of citric acid, amylases, actinases, proteases, statins, which are used for treatment of cardiovascular diseases, tachydiestase used in infant food and also used for the production of a fermented food called koji. Are all Aspergillus pathogenic? There are some species which are pathogenic and they are pathogenic under certain circumstances. Usually 100 spores they are inhaled by humans and animals daily, but they don't induce an infection. In certain cases, they may cause allergic reactions which are quite severe, and Aspergillus causes respiratory opportunistic fungal infection known as aspergillosis, not in all the individuals but usually in the immune-compromised individuals. Structure of Aspergillus. It has the septate hyphae. There are the cross walls called septum. The foot cell is generally L or T-shaped. The vegetative cell, foot cell, it bears the erected hyphae which is called conitiophore. The conitiophores get swollen to form the vesicle. This vesicle bears the elongated structure called phyllates. The round spores they are called conidia. There can be uniseriate Aspergillus or it can be biseriate depending upon the layers of the phyllates. In the uniseriate there is a single layer, while in the biseriate there is a double layer of the phyllates. These phyllates they are also called medullae. Characteristics of Aspergillus. They shows the culture characteristics. The Aspergillus fungus growing on the culture medium. It shows white, yellow to green colored colonies. The staining characteristics of Aspergillus, in which the hyphal structure, the septate hyphae and the conitiophore and the conidia they are shown. Pathogenesis of Aspergillosis. The Aspergillosis is acquired by the inhalation of the conidia, the spores. The spores they enter in the respiratory tract and they attach to the epithelial cells. In the respiratory tract, they are taken inside the lungs and usually they are engulfed by the phagocytic cells. If they are not engulfed by phagocytic cells, then they get colonized in the lungs and they may invade the lungs and evade the killing by macrophages. Then these spores they disseminate to different sites in the body. The clinical features of Aspergillosis. The Aspergillosis produces asthma-like allergic bronchopulmonary disease where there is difficulty in breathing and it may develop into permanent lung scarring. There can be intercavitary aspergilloma, also commonly called fungus ball. It can cause invasive pulmonary aspergillosis, in which the patient experiences fever, cough, dyspnea, and hemoptysis. Endocarditis, it can lead to the heart failure, shocks, and cardiomyopathies. There can be the cutaneous form of aspergillosis also, in which on the skin there are the septicemic infection, and it also causes the wound sepsis. Aspergillosis has been found in the patients of the COVID. There has been the reported cases of the black, white, yellow, and green fungal infections in the COVID patients. Out of this, the green fungal infection is the aspergillosis. The first case of green fungal infection in the COVID patient was reported in India on 15th June 2021. The COVID patients who are diabetic and under steroid therapy are at a risk of getting the various fungal infections. Lab diagnosis of aspergillosis. The specimens can be sputum, bronchial wash, tracheal aspirates. The first approach is the direct microscopy. The clinical specimen is taken on the slide and the QH wet mount is prepared. Under the microscopy, the non-pigmented septate hyphae with repeated dichotomous branch can be seen. The second approach is isolation and culture can be carried on the medium SDA Severod's dextrose agar. Aspergillus fumigatus produces fast-growing colonies which are white, yellow, yellow-brown, brown to black or green in color. Detection of antigen galactomannan can be an approach. This antigen can be detected in the serum sample of the patients. With the serological approach, in the patient serum sample, the IgE antibodies can be detected which are specific for the Aspergillus antigens, and IgG precipitants can also be detected. There are some questions for you. You can answer in the comment section. The question number 1. Name the pathogen causing green fungal infection in COVID-19 patients. Question number 2. Name the pathogen causing black fungal infections in COVID-19 patients. Question number 3. Name the pathogen causing yellow fungal infection in the COVID-19 patients. Write your answer in the comment section. You will get to know whether you are correct or not. I will respond to each and every comment. The treatment of aspergillosis. Various antifungals they can be given. Voriconazole with amphotericin B. Aerosolized liposomal amphotericin B can also be given. For the treatment of the allergic aspergillosis, corticosteroids or disodium chromoglycate can also be given to the patients. Prevention and control of aspergillosis. The exposure to the spores should be prevented. The transplant immuno-compromised, and the patients who are on ventilators, they should be isolated. A prophylactic low dose of amphotericin B or itraconazole for the patients who are at risk should be given. That is all about aspergillosis. Thanks for your watching.